Hi, everyone, and welcome back to High School of Mathematical Oncology's YouTube channel. Today, I'm very excited to introduce our guest, Dr. Jana Gevertz, a professor of mathematics at the College of New Jersey, where she specializes in mathematical biology, focusing especially on cancer modeling and immunotherapy. She received the Society for Mathematical Biology's Distinguished Service Award for her major contributions to the field. Dr. Gevertz, thank you so much for joining us today. To start off,
I think,
what
yeah.
first, uh, sorry? Oh, okay. Yeah. Again, thank you for joining us today. To start off, what first inspired your interest in mathematical oncology, and were there any pivotal moments in your career over the past 20 years that have shaped your path and the way you approach your work?
Yeah, there's sort of almost two separate answers to that question. So I'll start with what got me interested in mathematical oncology to begin with. So when I went to college, I had absolutely no idea what I was going to major in. So I know some people go and they already have a career in mind. I didn't, but I knew I really liked math and biology. I was not really aware that there was much of an intersection between the two fields. So I thought I had to choose between them. And after going back and forth between choosing the two, I finally landed on mathematics as my major. And then in my junior year, I found a class called Differential Equations in Biology. And I was like, okay, that's cool. I'm going to take that class. At the same time I was taking that class, my dad was diagnosed with cancer. He had a colleague 20 years younger than him given the same cancer diagnosis. They went through the same treatment protocol. My dad ended up being what you would call a super responder to the treatment. There was no evidence of disease after he went through one round of chemotherapy, and this was stage 3 cancer. And they kept treating him for a little while longer because that was such an unusual response. His colleague, who was 20 years younger, went through 8 rounds of the treatment and ended up dying a year later. And then to me, this question just like couldn't leave my brain, which is why are two people with the same diagnosis and the same treatment protocol responding so differently? And it was that question that was gnawing at me. And I knew I was never going to answer it by becoming a medical doctor, but I saw mathematics as a way to possibly try to really start to understand the heterogeneity you see in treatment response. So that was sort of like the culmination of events for me that really brought me to mathematical oncology.
Yeah, that's a such a powerful story. It's inspiring to hear, and I'm yeah glad your dad got
Thank
better.
you. I mean, of course, we're the lucky ones, right? That, he's still
Yeah.
here 25 years later is amazing. I think in terms of how I approach work over the career, the major, I think, change was when I first entered the field, models were very theoretical in nature. Like you would have a framework, you'd analyze it or you'd simulate it, and you'd make some broad qualitative predictions, but there was not a big connection to actual real world data and trying to connect the model to the data and use the model to actually impact how we approach running experiments or treating cancer. So the big evolution I've seen in the field since I've entered it is, right, we've moved away from strictly theoretical models, but to models grounded in data, models that are hopefully going to one day and are starting to some extent, actually impact how we treat patients.
Yeah, so it's really interesting, like mathematics and biology sort of converged for you, as well as
Yes.
how math has gone from like really theoretical to like there's like a lot of papers being published about how treatments could be used to help
Yeah.
patients and all of that. So I'd like to dig into how you actually build mathematical models.
Yeah.
So when building a mathematical model, how do you decide which aspects to focus on, such as balancing the need for simplicity in a model with the biological complexity of cancer?
Yeah, I mean, you're already asking a great question, right? Because the truth of the matter is, cancer is so complex. And if you try to model every single component of it, you're going to end up with thousands of variables, hundreds of thousand parameters in the model, and the model becomes completely intractable to analyze. You're never going to really be able to learn from it. But if you go too simple, you're not going to have enough details in it to learn anything meaningful either. And it's this constant trade-off between the complexity and the simplicity of a model. My answer, which sounds easier when I say it than it is to actually implement, is you need the model to be complex enough to answer your question, but not more complicated than that. So to try to give like an example, I have a few different models where I've looked at the same drug. So it's an immunotherapy, so it tries to sort of wake the immune system up to actually recognize the tumor as foreign and target it. And in one question, I was interested in just what's the optimal way to combine this drug with another drug. And for that model, I didn't really need to know the details of the mechanism of action of the drug. I needed to know what came out of it and the impact of combining with the other drug. In another study I did with the same drug, I was interested in understanding something about the mechanism of how the drug works. And the model I had used prior was not detailed enough to get to that. So then we had to add in the complexity because the question warranted the complexity. So that's sort of always how I try to approach it, right? Enough complexity for your question, but try not to go beyond that. And also enough complexity given the data you have available. So for instance, my sort of working approach is if I don't have data on how the tumor evolves spatially, but I know how it evolves in terms of its size and time, I try not to build a spatial component into my model because I don't have the data for that.
Yeah, that approach makes a lot of sense. Just focus on exactly what you need to answer the question.
And of
And
course, the challenge is you don't always know
yeah,
exactly what you need, but
understand.
that's what you try to look for.
Yeah. And then that like it's still complex, but like at least it's more understandable as a result.
That's exactly the balance we seek.
So in your work, you've mentioned concepts like parameter identifiability and sensitivity analysis. What do these terms mean in practice, and how do they help you determine whether your model is reliable or needs adjustment?
Again, it is really a great question. So this comes down to the idea that when I have a model, I always want to validate that that model does something I see in the real world. So if I'm trying to predict how a tumor responds to a specific drug, I want to make sure I calibrate my model so it describes that data. So what does it mean to calibrate a model? So a model is a set of mathematical equations, but in the equations are a bunch of what we call parameters, which are just constants that you can set the value of so the model output looks like your data. And then the question becomes, how sensitive is your model predictions to the exact values you pick for those parameters? So for instance, you might have the case where there's a parameter in your model that controls how quickly the tumor grows. And if you change that parameter just a little bit, maybe you don't describe the data well at all. You have other parameters that you might be able to change a lot in your model and still describe the data equally well. So the idea of sensitivity is how confident do I need to be in the value of this parameter to describe the data? On sort of the other side of the coin is this idea of identifiability. A parameter would be called identifiable if there's enough data for me to know with confidence what value that parameter needs to be for the data set. If the data doesn't give me a lot of confidence in the value of the parameter, I would say that parameter lacks identifiability. I can't figure it out from the data I have. And this can sort of be a double-edged sword. So if a parameter doesn't have identifiability, you don't need to know it very well to describe the data. You could describe the data well, no matter what you do with the parameter. But the problem then becomes if I then want to use the model to make a prediction about another scenario, what if I combine this with another drug? What if I want to change the protocol? If the parameter wasn't identifiable, maybe I could take two values of the parameter that perfectly well describe my training data and make a prediction, and they predict totally different things. So when
Yeah,
you lack identifiability, you run the risk of not knowing the right value, the true value of the parameter, and then therefore not being able to trust your future predictions because of that.
that's really eye-opening how a tiny change in a parameter value can like just shift the model so much about how well it represents the real world.
Right, and it's tricky because if it's a sensitive parameter, you might change it a little bit and no longer fit your training data, but at least you know the parameter with confidence because your parameter really has to be that value. And then I know when I make a prediction forward, I can really trust that parameter.
Yeah. And like
But the flip is you can't always measure a parameter well. So what if someone can't measure it for you well? Then you don't want to be overly sensitive. So these are tough questions.
And I guess cancer cells don't really make it any easier since they constantly adapt to their environment.
Yep, it's such a good point. Not only do cancer cells adapt, cancer itself is very, very heterogeneous. So you might measure a parameter from a biopsy sample, say, right? And then you look at what happens from that sample to measure a growth rate or a response to treatment. But if you possible that if you biopsied a different region, you would have got different parameters out of it because of the heterogeneity you see within a tumor too. So yeah, doesn't make it easy.
Could you explain the difference between drug-induced resistance and pre-existing resistance in cancer cells? And
Sure.
yeah, I guess also how phenotypic plasticity adds more difficulty to cancer treatment.
Yeah. So it used to sort of the paradigm that we used to have for resistance to a drug, and you can think of it as chemotherapy, but really any therapeutic, was that there's a small population of cells in the tumor that before you even gave the drug, for some reason had properties that made it resistant to that therapy. So what would naturally happen then when you give a drug that can kill most cancer cells, the tumor is going to go way down in size because you kill off all those sensitive cells. But you had these few in the pool that were never going to respond. So eventually, as the sensitive cells disappear, those cells are able to start growing and growing, and they don't care about the drug and they take over. Now, in theory, if drug was resistance was only pre-existing, if you could find a second drug that could hit those cells, you'd pull them down too. And maybe you would even need a third, but eventually you'd probably find a drug that hits all of those cells. Now it might become too toxic, a pool of drugs, but in theory, you might be able to overcome the resistance. It's being better appreciated now that cancer cells are quite what we call phenotypically plastic, like you said. And what I mean by plasticity is that they could actually change their behavior in response to stresses in their environment. And drug is an example of a stress, but low oxygen or low nutrients are other examples of stresses. And what cells do is they don't genetically change, but they change which proteins they actually express because of the stress in their environment. And by changing the proteins they express, they change their behavior. So a cell that inherently had no reason to be resistant to the drug, due to the stress of the drug, might start to change the way it behaves. So therefore, the resistance wasn't just selected for, it was actually created by the treatment. And therefore, you can't just throw another drug at it and think you're going to change it because it happened because you put the drug in to begin with. So then this notion of how to overcome the plasticity or what we're calling the induction of resistance becomes another level of challenge in designing treatment protocols.
Yeah, it's wild to think how cancer cells can just reprogram what proteins they express in real time to
You know
survive
what?
the
What's
environment.
crazy about it is really what cancer has done is hijacked all these mechanisms we tend to really appreciate in our normal cells, right? During development, you need your cells to be quite plastic, right? You need them to be able to shift what they do in response to changing gradients of chemicals. During wound healing, you want cells to change their behavior so they respond to the wound, right? These are normal mechanisms that cancer cells are hijacking to their benefit.
Yeah, it's like I go on YouTube, like the Kurtzagot in a nutshell videos about cancer.
Yeah,
It's insane how much a cancer can do to adapt
I know
against.
they almost feel smart in that regard. Yeah.
So in the clinic, doctors usually only have access to the total tumor size.
Yeah.
With such limited information, how would you figure out what's actually happening inside the tumor, such as the number of drug-resistant cells and drug-sensitive, and how they're sort of adapting to
Yeah.
the new environment?
I know and this is a really difficult question. So the biotechnology is developing to the point where if you were able to apply all the tools available to data from a patient, we do have ways to do genetic barcoding and things where you can start to really track evolution of different cells with time. We have mechanisms, ways now that you can start to identify rare cell populations that have rare behavior. But the problem is these tools are never available to the average patient, right? They are expensive. They are not part of standard clinical care. So I think a real question that we're trying to get at that we're actively working towards is sort of what is the minimal amount of data we do need to be able to get from a patient so that we could build predictive models that have enough information in them that we can then use the model and make a confident prediction. But this is very much work in progress. I don't think anyone has gotten there yet. But as biotechnology develops and more data can be collected, I think math has a real role in saying, all right, you have all these tools. What are the most important tools to apply and when? So that way I have the minimal amount of data I need to make good predictions for your patient. So we have a long way to go till we're there, but that's sort of where I see it going.
That's a really cool detective story, just like using whatever information you have. And maybe like in time, the biotechnology will
Right.
become
Costs come down
bigger.
a lot, right? The more, but right now a lot of these things are still really experimental in that they're literally applied in labs that don't do clinical research, but in labs that work on, right? They might have xenograph models where, you might have gotten a patient sample, but then they're just putting it in a mouse and they're looking at, things in a mouse, or they have preclinical data where they're literally in a petri dish. Like it hasn't translated yet to the level of a patient.
Yeah, so going from like the lab to directly helping patients,
Yeah.
can I ask about how models improve treatments, such as how does mathematical optimization identify the best possible treatment strategy for a patient? And
Yeah.
could you share a real example from your research?
Sure. So let me sort of give like the theoretical ideal of this, and then we can talk about sort of practical implementations of it. So theoretically, the challenge is that imagine you have a patient in front of you and they're getting a cocktail, we'll just pick a number, three drugs. Now, if you have these three drugs, you need to decide for that patient not only when to administer each of the three drugs, but how much of each of the three drugs to give, right? So it's a question of both dose and spacing. And do you give the drugs together? Do you give them apart? What is the optimal way to do this? And if you tried to think of running a clinical trial to test all the possible combinations, there's way too many combinations to ever actually test in practice. So this is where a predictive mathematical model can come in. If we have a model that we believe well describes what we see happen in patients, we can use mathematical tools to try to optimize the way to give these treatments. So that way it's the model that performs the optimization, not people where you're, experimenting with people's lives, but then you can take what you've learned from the model and bring it back to the clinic. So that's the ideal. There's too many ways to possibly think about how to dose and schedule drugs. So you do it in the model and you bring it back to the patient. So that's what we'd like to happen. The challenge then becomes, how do you do this in practice? Right. So I would say the field is still working towards actually translating many mathematical predictions into clinical, right, application. But we're starting to see some clinical trials where this is happening. These tend to happen at places where the mathematicians and the clinicians work actually in the same hospital. So for instance, you see trials run at Moffitt Cancer Center or MD Anderson, where the model-based decisions are actually being used in the clinic to inform how they actually dose and schedule. And it could be as simple as, the patient comes in every month for an image. You get the data out of the image and you use that data and the mathematical model to say, should I wait on giving drug or does this person need drug today? Or how much drug should I give today based on what I've seen over the last month of imaging? So models are starting to come into play to make these decisions. We still have a very long way to go until it's a regular integration. Most people right now, when they go to get treated, mathematical modeling is not influencing their treatment.
Yeah, it's amazing how math can help find better treatment options so much more safely and then just like sort of predict how the patient will respond.
And
And then,
it also requires getting to the point where clinicians too trust that these models can say something that's both useful and safe for the patient, right? I mean, they're in a very difficult situation. If a model says, hey, actually, don't give any drug this week, the patient will be like, wait, you're not treating me this week? Don't I need my drug? But the model might actually say, you are better off not having drug this week. And that takes time, right? To say, or you should take half the dose you took last week. Why should I do that? Don't I want to kill my cancer, right? So it's not a it's not an easy translation from what the model says to what the clinicians have to actually do with an ill patient in front of them.
Yeah. And I guess to sort of build that trust, it's like really important that continuous like experimentation of real cells and tissues happens as well.
Exactly.
So, yeah, how do your mathematical models interact with lab experiments such as single cell analysis?
Yeah, so I actually, none of my work has gotten to the detailed level of the single cell. I'm much more interested in tissue level response. So how the tumor actually responds to whatever treatment protocols, you're using, or the evolution of resistance within the tumor mass. There's so much great single cell data coming out right now that can inform all of these things, right? They factor into what drives resistance, what drives treatment response. The people who tend to look at those models are looking at what we call multi-scale models, where you bridge what happens inside a single cell with what happens at a much higher level, the tissue level. There's people who do beautiful work in this area. It's just not the focus of what I've been doing.
Yeah, that sounds really impressive. So, on a scale of like a tissue scale model, how does like how would that sort of inform a biological study, the two of them in combination?
Yeah, so for us, what we're typically interested in doing, so let me give you an example. So we had some data about the way two drugs were interacting with each other in two different non-small cell lung cancer cell lines. So one harbored a particular mutation that the other didn't have. And those two cell lines responded very differently to the same combination therapy. And they had done sort of one standard protocol for each of them. So once we're able to take a model, calibrate it to what they saw happening to the bulk tumor volume in those two cell lines, we can sort of use our underlying mathematics and optimization techniques to say, all right, for patients that have this mutation, what would we recommend is the better way to treat these patients? And what if you don't have that mutation? So we try to take that sort of bulk data on here's how the tumor changed over time if you have this mutation or not, and use it to make different recommendations based on the model of how much drug each of those patients would actually need. But we
Mhm.
never looked at, even though we know one cell line has a mutation and the other doesn't, we never looked any more detail at the cell lines themselves.
I'm not sure if like this method is completely the same as a virtual clinical trial,
Good question.
but yeah, but like quick question, what are virtual clinical trials and how do they improve cancer treatment slash create and model virtual patient populations?
So maybe let's start with a clinical trial, right? So when we think of a clinical trial, we think of when a bunch of volunteers, and these will be some people with the disease, sometimes people without a disease, are recruited to test the efficacy of a new therapy, the safety and efficacy of a new therapy. So the challenge of clinical trials is, first of all, they rely on people volunteering for them. And then they're putting an experimental drug in their body. This is not only a very time-consuming process, it's very costly to recruit people, treat people, give them medical care around sort of the treatment that they're testing. So there's a lot of limitations on sort of not only the time constraints and the cost of clinical trials, but also who enrolls in them. So historically, we've seen clinical trials tend to be in the United States, tend to be way overrepresented with white men. But you would want a sort of diverse representation of people in your clinical trials because we know people of different backgrounds, genders actually can respond very differently to the same drug. So virtual clinical trials step in and try to say, if I have enough data from a clinical trial, can I then build a model that represents what I see happening in the data, but then run the trial on the model itself instead of on actual patients, where different patients are just represented by different parameterizations of your mathematical model. And because it's much easier to change parameters in a model than it is to recruit very diverse patients to a real clinical trial, what you're able to then do is try to understand the effects on a more heterogeneous population through the equations instead of doing it through actual people.
That's incredible to hear how virtual patients and like the virtual clinical trial, you're able to use like pre-existing data and just sort of build off of it. Though I guess it would be important to have the like a pretty good understanding of the parameters for that.
Exactly. It's sort of a perpetual challenge in doing this is what is a realistic parameterization for a patient? Because if you bias your parameters too much by the people who are in your pool, you'll never get the diversity of the people who are not in the pool. But you can't go so crazy on varying your parameters that you have parameters that don't represent any realistic human being. So it's a challenge to sort of say what is a realistic virtual patient. But if you can design a realistic pool of virtual patients, then you can run the trial effectively on your computer with these equations instead of with real people actually getting the drug.
Yeah, and that'd be like it's a lot easy, like cheaper, faster,
It's
which
cheaper,
is like always a
faster,
problem.
and safer because you're not, right, exposing all these people to an experimental therapy.
So, as we're wrapping up the interview, I'd like to ask what foundational skills, whether in math, coding, biology, should
Yeah,
high school students or early college students focus
yeah.
on if they're interested in mathematical oncology?
Yeah. So what I would say is you sort of hit on the fact that it's very interdisciplinary, right? So there's not just one skill you're going to need to come into this. And over time, these skills could all be built up. But what I would say is, in the three areas, foundationally, you need to have some sort of interest in the biological question, right? If you're not sort of motivated to understand the biology of cancer and how people are, what different treatment protocols are out there, it would not be the field for you. But if those questions are of interest to you, your foundation is already there. Skill wise, in terms of mathematics and computer science, the main mathematics we use are called differential equations. And if you've taken a course in calculus, you've already learned the foundations of differential equations because differential equations are equations that involve derivatives. And derivatives are what everyone studies when you take your first course in calculus. So you would want to start by getting a foundation in calculus so you understand derivatives, you understand integrals, and then as you build up that foundation, you'd be ready for a course we call differential equations, which in some places is called like calculus 4, like you get there in your calculus sequence. You also will definitely need computer programming skills. It is not super important which language you first learn in, because once you learn the basics of programming, working in many different languages sort of gets you to the same place. So I happen to use a program called MATLAB, but a lot of people now use something called Python as their language. If you learn in Java in high school, that's also okay, right? But just understanding how to build algorithms and sort of instruct a computer to follow a set of directions you want it to follow is a really important skill.
Yeah, that's really helpful advice. And yeah, I guess you also really showed how interest into the disciplinary learning is really important to the field of mathematical oncology. So for most of our audience, it's a long road ahead for
Yeah,
research.
yeah,
And I guess research isn't always easy. There will be failures and all that. So how would you support students through setbacks? And what advice would you give to someone who wants to use math and science to make a meaningful impact in cancer research?
yeah. I'd start by saying one of the things I always see people struggle with when they enter an interdisciplinary field like math oncology, because it's so interdisciplinary, you feel like you need to know everything. You feel like you need to know all of math, all of statistics and data science, all of computer science, all of biology, and no human being is ever going to gather all that knowledge, right? So one of the things is you sort of have to accept when you're interdisciplinary, there are going to be things you know well and other things you need to lean on other people for, your collaborators, your mentors, and that's okay, right? That you're not going to get yourself 5 undergraduate degrees just to become a mathematical oncologist. There are many pathways into this, right? If you look at who does mathematical oncology now, there are people whose undergrad training was in math and bioengineering, in computer science, right? We come from all different places to get here. So there's not one way to do this, first of all. Um, but second to your question of sort of persevering through difficult research, I think you've already said the most important thing. You need to sort of know when you start that doing research is not doing homework for a class, right? That we don't know the answer already when you start. So if you're a student in my class and you struggle on a homework problem, you know if you come to me, I'm going to be able to answer the question, right? Because I assigned the homework. But in research, there isn't always a good answer to the question. So sometimes I can give a student a project and we can spend 6 months on it and then realize, wow, we can't find a good answer, or maybe this wasn't a good question to begin with, or we should have asked this question instead. And unfortunately, that's sort of the nature of research, but it's healthy too. If we were asking something where the answer was almost obvious before you start, that's not research, you know? So you just need to know not to be discouraged, to understand it's par for the course, but to have tools to say, how do I work through those roadblocks? If we found we've asked a bad question, how do we pivot to a good question? Because often you'll find the good question once you realize why your question was not the best one to ask. So patience, perseverance, understanding of yourself, understanding mentors are all sort of ways forward.
Yeah, Dr. Gevertz, thank you so much for such a thoughtful and insightful conversation. You've given us an incredible look at how math can be used to fight cancer, from theoretical modeling to patient-level impact, and how students like us can begin building the skills to contribute. For those of us watching, if you're excited by the idea of combining math, biology, and coding, I highly encourage you to explore this field further and check out some of Dr. Gevertz's work. Whether you're interested in programming, biology, or solving equations, as Dr. Gevertz says, there's a place for you in mathematical oncology. And thanks again, Dr. Gevertz, and thank you everybody for tuning in.
Yep, thank you. Thanks, Isaac. I really appreciate it. It's great that you're doing this channel. I think, to try to reach people early and let them know this field is here and how they can get into it. I would have loved to have known this. So I think it's great that you're doing it.
Yeah, thank you so much. Stopping recording.